All right, so for this audio lecture, we're going to be looking at mechanisms of genetic variations. We're going to focus on a couple different aspects. So first, mutations, we'll talk about DNA repair, and then a good portion and what is kind of unique from like a genetics class that's focusing on eukaryotic organisms is going to be horizontal gene transfer, which includes congregation, transformation, and transduction. So a lot of this in the mutations is probably going to be a review from a genetics course that you might have taken or even an intro bio course. So with mutations, we want to keep in mind that we can change base pairs in the DNA from what was inherited, and that would be recognized as a mutation. Okay, point mutations are the most common, so where we have a single nucleotide being either added, deleted, or altered. Okay, we can get larger mutations. These are less common, but this is where we have an insertion, deletion, inversion, duplication, translocation of a collection of nucleotide sequences. So this could be like a whole entire gene being inserted in and then disrupting another gene, right? And there's a lot of other examples that, again, if you were to take a genetics class, you would go into much more detail than what we are going to be doing in this kind of survey of mechanisms of genetic variations. We want to keep in mind with mutations, they can be spontaneous or induced. So with spontaneous mutations, there's no external agent that is driving it. Okay, so it's a little bit more from an error in the machinery. So when we're looking at DNA replication, keep in mind we have a number of enzymes that are involved. Those enzymes are not 100%. There are proofreading mechanisms in place to make sure errors are caught. But again, even those are not 100%. And so if we're thinking about eukaryotic organisms as we age and our cells have gone through a number of divisions which require gene replication, it is more likely because at each time there is a chance that there could be a mutation. So over time, those mutations could add up and then they could lead to where we actually see a change in phenotype of those cells, which then would lead to like cancer or such. Okay, so we also want to keep in mind that we also have mechanisms that allow for mobile gene elements to be active. And so when we look at like transposons or these jumping genes, that would be considered a spontaneous mutation because it's like within the cell. So again, anything that's coming from within the cell is recognized as a spontaneous mutation. It may be that someday in the future that some things that have been classified as being due to spontaneous mutations might get changed and being recognized by being induced by outside forces. But again, just keep in mind that at this current time, when we're talking about spontaneous mutations, we're talking about there's no external agent that has been identified that causes. It's just an error due to the machinery that is in the cell, responsible for DNA replication or normal processes like jumping genes. With induced mutations, this is where there's been some kind of outside agent that causes damage to the DNA. So this is exposure to some kind of chemical or UV exposure. We know certain chemicals can damage DNA. We know UV exposure can damage DNA. So when that DNA is damaged, there are repair processes that are supposed to fix that. But again, keep in mind, repair processes are not 100%. And so if there is high level exposure and multiple breaks in the DNA, it's going to be more likely that mutations would arise. So sometimes in the fixing of the DNA, we can get base pair oligos. So they're structurally similar to the normal bases, but they are not. So again, this could lead to a mutation. We can get DNA modifying agents, so they can alter the base pair, causing there to be a mispairing. Intracellular agents, they disorient the DNA to induce single nucleotide base pair insertions or deletions. We want to keep in mind with mutations, there's different types of effects. So we within a population, the most prevalent form of the gene is known as wild type. So that's the quote unquote normal. So variations from the normal would be a mutation. So a forward mutation takes the individual from the wild type normal to a mutant form. It is possible that you can have a mutation that's reversal, and it actually brings the person back from the mutant form to the wild type form or their offspring, right? And we can also have suppressor mutations. This is why the wild type phenotype is restored by a second mutation that's at a different site than the original mutation. So the example I mentioned in class is that, and this is a eukaryotic example, but we as human beings actually, lactose intolerance was wild type. That was the norm. So mammals stop expressing lactase, the enzyme that breaks down lactose, the sugar. And so it is normal to lose the ability to express that enzyme, which means then you can't consume dairy products or things that have lactulose in it. It upsets your stomach, causes gas, and a number of other unpleasant things. So what happened was there was a mutation that formed that instead of that gene being turned off, it continued to be expressed past the normal age of like around 6 to 9, that it normally would turn off, and then individuals would start weaning themselves for humans. Okay. And so with people having the ability to continue to express lactase, that meant that they were able to continue to consume high-fat foods that so happen to contain lactose, and that gave them a higher likelihood of being able to survive really tough times like winters. They had more insulation on them, fat, right? And then that also gave them the higher likelihood of being able to have reproductive success, so fitness, right? And so that meant they passed that mutation on to individuals. And so again, especially in certain areas, you see the wild type expression is that you continue to express lactulose into adulthood. Now, within our population, we have some people that, whether it is because of the genes that were passed on to them or whether there was some kind of reversal mutation or suppressor mutation, they are now lactose intolerant. And so now we have a whole market of products that do not contain dairy or do not contain lactose so that those individuals can process them. Just keep in mind that being lactose intolerant is very different than being like allergic to dairy. Those are two different things. One's an enzyme, and I'm going to say deficiency, but again, it's just actually returning to your normal state that human beings had generations and generations and generations ago, where being allergic to something is the immune system is responding. So we want to keep in mind if the mutation happens where it is the protein coding portion of the gene, that means it could alter the protein structure, and we know that how the protein structure is important for the protein to function. So if we are changing the amino acids that make up the protein, that can affect whether the protein can function or not, and there are different types of mutations based on whether there is a change or not. And how that change occurs, and we'll talk about that in a moment. If there are changes to the protein coding gene, that mutation is named accordingly depending upon what type of mutation it is. In this table from the textbook, you can see that the different types of mutations. So again, within the forward mutations, we have different types. So you can see here on the examples where the first line is the nucleic acid sequence, and then the next line is the amino acid sequence. So you can see the first one is the normal, the wild type, so there's no mutations on it. The next line has the silent mutations. So you'll notice that there is a change to the nucleotide, right? So is a mutation there, but it's silent because there's no change to the amino acid sequence. So if you compare the amino acid sequence between the non-wild type and silent, there's no changes in it, right? So it's not going to change the protein and its function. Now, it is possible it could change the amount of protein available, because if it has changed the transfer RNA and there's a change in the abundance of the transfer RNA, and this is where sometimes we call them silent, but there may be an effect on the amount of protein that's actually expressed, just to full disclosure. You can see with our missense mutation, there's a base pair substitution, and so this leads to a change in the amino acid that is present. So in that case, given the property of the amino acids, if those are very different than the original amino acid, that will dramatically change the function. If the amino acid that it's changed to has similar properties, it may not affect the function as much, or even at all. So if you remember back to your amino acids from IntroBio or CellBio, amino acids have different properties like nonpolar, polar, and their side groups kind of determine that. So if you change a polar amino acid to a polar amino acid through a point mutation, like a missense, again, that may not have too much of an effect because you're going polar to polar. If you were to switch it from polar to non-polar, or vice versa, that would have a dramatic effect, right? Because you're changing not only changing the side group, but changing the properties of the side group. And we know the side group can affects how the protein's going to fold, whether it's going to be found transmembrane or whether it's not, whether we have alpha helixes and beta sheets, because we're going to change the secondary structure, right? With nonsense here, you can see how this base pair substitution actually codes for a stop codon. So this is actually going to stop the expression of the protein early. And so we could have a whole domain. It could be a substrate binding domain that is like removed because we have a stop codon. So it likely means that protein is going to be non-functional. Now, it is possible that stop codon, that mutation's at the very end, and maybe that just changes that signal sequence, right? So if it's in the last 4 amino acids, and we're talking about the KDL, which is a signal sequence that keeps the protein in the ER, then that might change it so it actually gets secreted. So again, nonsense, it depends on where that stop codon is, how it affects that protein. And then frame shift, that's where we've added or deleted a nucleotide. So that actually changes the codons that are created from that point on, right? So codons, which are determining which amino acid is used, is reading those 3 base pairs. And so if we remove or add one, there's a shift in those 3 that we are reading. And that would change the amino acid, and again, would change the protein. Okay. I'm not going to go through the additional ones because, again, this is not a full genetics class. This is just meant to be a survey, and you should have a general understanding of what reverse mutation and suppressor mutations are. It is the forward mutations that I expect you to be able to identify and match with descriptions and such. For some mutations, they are only expressed in certain environments. So those are known as conditional mutations. So under certain conditions, they're expressed, right? So if we had a mutation to the lac operon gene, we wouldn't actually know that unless there was lactulose present, right? So we talked about the lac operon last unit. And so we need to have lactulose present for that operon to be turned on and express those proteins. So we could have a mutation in there, and if there's never lactulose present in that bacteria's environment, we would never know. And that would be an example of a conditional mutation where under certain environmental conditions it is expressed. Okay. Autotrophic, that is where there's an essential, they're unable to make an essential macromolecule such as an amino acid or a nucleotide. So it is an example of a conditional mutation, but it's specific to it's related to this essential macromolecule. Mutations can also occur to the regulatory sequences. So again, thinking about the lac operon, we had said how there's that regulatory portion that produces the repressor. And so if we have a mutation to that repressor, it's not going to be able to express the repressor, which means it would always be turned on. So we'd always be producing the enzymes that were needed in order to metabolize lactulose, whether lactulose is present or not. Not very energy efficient, right? We can also have mutations to the tRNAs and RNA, ribosomal RNA genes. So if those are involved in protein synthesis, so if we have mutations to those, that can actually disrupt protein synthesis across the board, right? So if we affect our ribosomes, then we can't assemble and make our ribosomes correctly. Well, they can't bind to the message RNA, even if it's made, so we can't make proteins. And again, it's not going to, mutations to tRNA and ribosomal RNA are not going to affect just one protein. It's going to affect all protein synthesis. So how do we detect mutations in with microbes, right? So obviously with human beings, we can do genetic screening. If it's a phenotype, we can make observations that way. With microbes, because they're small, it's a little more difficult, right? So sometimes we can make observations of phenotype changes. So there might be some kind of behavioral or phenotypic change. So it might be something where the bacteria doesn't create biofilms, or the bacteria does now create biofilms. On a media plate, on an agar plate, that might look like where they're piling up on top of each other and creating a ridge. So again, some phenotypes we might be able to observe. We might be able to look at them underneath the microscope, and we can observe whether they express flagella or don't express flagella. Other ways to do this is by doing replica plating techniques, and that's what's shown in this image here. So for the replica plating technique, what you would do, this is used to screen for mutants, especially conditional mutations. And so we have our master plate that has the complete media, everything that the bacteria would need in order to grow, regardless of whether they're a mutant or not. And then what you take is this replica block that has a velvet surface on it that has been sterilized, so it gets autoclaved in between use. So when you press that down gently, you don't even press it, you just tap it onto your master plate, some of the bacteria transfer onto that velvet surface. So then you tap that down onto your replica plates. You would have multiple replica plates. Some of them might have different media in them. So you'd have some that have complete media, and then you'd have some that would have some kind of media that is either lacking or has some kind of component. So if you're looking at something like antibiotic resistance, one of your replica plates would have media that has the antibiotic present, so that you could determine whether they are resistant or not, right? Or maybe we're looking at, synthesis of a certain, or metabolism of a certain amino acid, or it could have, one media could have lactulose and one media maybe doesn't have lactulose. So then after you incubate those, you observe the growth. And so you have your master plate and also your replica that has the complete media to compare it to. And then you would look at your media that has that condition in it that should induce the mutation, so you can see. So then you would see, where do you not have growth? You could then look at your master plate and pick up the colonies that were growing on the master plate but not on your conditional media. And then you could do additional screening. So that's where you might then actually sequence their genome to determine where the mutation is. You could cut out the gene that you think it is through restriction digest, or do PCR to amplify that gene and just sequence that portion. Or you could do additional digest that target the area that you think there might be a mutation to determine whether it is present or not. So a number of different ways that you could determine that. Okay. So this is kind of the classic microbiology way to determine. Obviously, you could also just isolate the bacteria and do sequencing to determine whether there's mutations occurring. All right, so next we're going to talk about DNA repair. So again, keep in mind that when there are errors either through synthesis or spontaneous errors or induced errors, there are repair mechanisms that are meant to correct that. So there are proofreading mechanisms that correct those base pairs that are made during replication. Those errors get corrected by DNA polymerase, but again, keep in mind DNA polymerase made the error to begin with if it was spontaneous, and so it's possible it could continue to make errors. We also have other DNA repair mechanisms. So one of them is where we have mismatch pairing, and so this actually cuts out portions. So there are enzymes that are going to cut the DNA that is newly synthesized, that's mismatch, and then DNA polymerase comes in, synthesizes the DNA, and then DNA ligase then ligates those ends. And the way that bacteria are able to determine and distinguish the old strain from the new strain is because they methylate the old strain. So the old strand is going to be methylated. The new strand lacks those methyl groups, so that allows the enzymes to be able to determine which is the strand that we should leave and which one needs to be repaired, right? So the repair is going to happen to the unmethylated, the new DNA. So that's what you're seeing in this image here, that there's a mismatch that's been detected. So we have an incorrect base, and so at a cleavage site that allows the DNA polymerase to bind to it, we're going to cut that. The incorrect information, which is unmethylated, is removed, and then DNA polymerase with DNA ligase comes in and synthesizes new DNA and then ligates that. So we have that correct pairing, as you can see at the bottom of that figure. We tend to think of methylation in eukaryotes as this like turning off of a gene and turning on, so how we can regulate why one allele is expressed and not. So I just want to point that out because this might be something new for you on how methylation is used in microbes and how it's a little different. It's another approach, a different approach than what is used in eukaryotes. So we want to keep in mind with mutations, they have to be subject to some kind of selective pressure in order for those mutations to truly matter, right? So each mutant form that survives then becomes an allele, an alternative form of that gene. So, when we're talking about different variations of a gene, whether you have a blue-eyed allele or a brown-eyed allele, keep in mind originally the blue-eyed allele is a mutant, right? And then when it becomes and survives generations, then we would recognize it as a variation of that gene. Okay. Also, the other way that we can create additional gene variability is through recombination processes. And so we will be talking about those in just a moment when we talk about horizontal gene transfer. The products that are formed by that, as far as like organisms being the product, those are known as recombinants. So before we talk about that, just briefly, with sexual reproduction, we want to keep in mind that's a process that eukaryotic microorganisms can undergo. Some eukaryotic microorganisms undergo asexual reproduction, right? But if we have sexual reproduction occurring, there's going to be new gene recombinations. So I always kind of joke the purpose of, sexual reproduction is solely for genetic variability, right? So you are going to have meiosis occurring. There's crossing over events between the sister chromosomes, so you get new combinations of genetic combinations as far as the alleles that are found on a single chromosome. And then we also have the fusion with gametes, right? So we have two gametes coming together, each contributing novel genes to that new organism. So that leads to new genetic combinations. So that process we recognize as vertical gene transfer. So where we're transferring genes from parents to the progeny. Vertical transfer also is happening in asexual reproduction. Genes are being passed from the parent cell to the daughter cell. The difference is there's no, in that process, there's no genetic variability that's coming up. When we're talking about asexual reproduction, it's just synthesizing, copying, and then passing it on, unless there's been some kind of mutation that occurs. So the question becomes, how do these prokaryotic organisms, specifically bacteria, increase their genetic variability? And the way they do that is through horizontal gene transfer. So horizontal gene transfer is a transfer of genes from one organism that is independent, mature, at an adult stage to another. So a stable recombinant will have the characteristics of both donor and recipient. So it's not a full gene transfer, it's a portion, a single gene or multiple genes that get transferred from the donor to the recipient. The donor keeps a copy of it, so they are replicating, or if the donor is alive, is living, they will keep a copy of that information. So they retain those genes, and then they pass a copy on to the recipient. In some cases, the donor is not alive, and so we'll look at these different variations. But this is very important for the evolution of different species. So if we're looking at expanding into an ecological niche, exchanging of genes with organisms that are living in that niche is beneficial. It increases our virulence, our ability to, the microorganisms' ability to survive that. So there are 3 different mechanisms that involve bacteria to create recombinants. There's transformation, transduction, and congregation. So in these, the genes can be transferred to the same species or different species. So we'll end up talking about these 3 processes in more detail in coming slides. So we have transposable elements. So these are segments of DNAs that move about the genome in a process called transposition. So what this means is that we have these genes that can jump from one location on a chromosome to another location, or even to different chromosomes. Okay, so these sometimes and historically were called jumping genes. So if you go back to old textbooks and you can't find transposons in there, look for jumping genes. It's probably there. So there's different mechanisms that can be at play for these. And the simplest one is that there's just an insertion of the sequence, and it inserts just right so that it does not disrupt anything. But in some cases, when these genes jump, they can actually disrupt another gene. They can also be inserted under a different promoter, so that can change their expression level. There are mobile gene elements that contain the genes for the enzymes that are needed for this process to occur. Okay. And if you saw my class announcement about that podcast actually talks a little bit about those mobile gene elements, and they were looking for those mobile gene elements to indicate whether these genes that they were looking at were able to jump or not, whether the organism could have jumped the genes themselves. So just to link that with some of the current stuff coming out. So in this figure, what you should be taking away from this is that transposase is one of the enzymes that's found in these mobile gene elements. So it's an enzyme that's needed in order to make genes jump. In panel B, you can see how there are copies of transposase, and they are located on both sides of that antibiotic-resistant genes. That makes it more likely that gene would be able to jump to another location. You'll notice flanking on both sides are these DRIR. So some of that are these, I had mentioned in class about the ratio of CG. So in there are these sequences that indicate that that is an area where there is mobility. So another term that if you're looking at jumping genes is pathogenicity islands. So organisms, bacteria can have these pathogenicity islands, and they tend to have these mobile gene elements located near them and on them, and they have altered CpG ratio, and they tend to, as the name and kind of hints to, the genes code to virulence, ability to survive, ability to be pathogenic, to cause disease. The island piece is that they can, they don't just float wherever, but again, they're part of these jumping genes. So there's a simple transposition. So it's kind of a cut and paste. So here we can see in this image where we have the transposase cleaving that gene, taking that gene to another location. So again, simple cut and paste. Replicative transposition is where the original transposon remains at the parent site, but a copy is actually inserted into a new location, which then means we have 2 copies of that gene. When another aspect of when we're looking at horizontal gene transfer is plasmids. So we had mentioned plasmids at the beginning of the semester, that bacteria have a single circular piece chromosome where they have their essential genes, and then they can also have these small pieces of DNA which are plasmid. They are also circular, and they tend to contain more luxury genes, genes that allow them to survive certain conditions, but not needed for every single day. So with plasmids, those are independent from the host chromosome. They can integrate into the host chromosome, and then they can also exit that host chromosome. So again, there's variations of it, but again, when we're looking at a plasmid as a plasmid, it is going to be independent from the host chromosome. So one of the chromosome plasmids that's looked at is the F plasmid. This one, we through research, we know it can undergo congregation. So it's known as a congregative plasmid. So it can transfer those copies to other bacteria through the pili. Just to show you what the plasmid looks like, it is going to have certain factors that are needed in order to form those pili, and then it would have additional genes that might code for antibiotic resistance or such. So back in 1946, researchers identified that genes were able to be transferred between bacteria, and so the way they did that was through the formation of this pili, which you can see here in this image. It's kind of like a straw-like structure, and allows for transfer from one direction to the other, from the donor to the recipient. So maybe, not knowing which direction it's going, this bacteria over on the left that has fembrii might have a plasmid on it that codes for fembrii, which can lead to increased virulence because you can attach to surfaces and tissues. So it would make a copy of that plasmid, and then it would go through that pili to the other bacteria. Original bacteria retains that because they've made a copy of the plasmid. The new bacteria now has that plasmid and whatever genes are coded on that plasmid. We now know that congregation can be bidirectional. So there are bacteria that are able to transfer when they are receiving a plasmid. They can make a copy of a plasmid and transfer to the other one. So unidirectional transfer is not the only, it's not always just in one direction, though it is more common. This image here just shows you a little more detail about the process. So you can see these 2 bacteria. The top bacteria has the plasmid. It has the ability to make the pili, so it will express those proteins, assemble, which we'll see in the next figure, assemble that pili, so they're making connections. It will then replicate the whole entire plasmid, and as that is replicating, that ends up going through that straw-like pili into the other bacteria. So now both the bacteria have that plasmid and the genes that are contained in that plasmid. And here you're just seeing the plasmid being created, and again, what's showing you on the right is just showing how that strand, as that is being produced, you're also producing the complement strand so that can go through the pili. On the left, you're seeing the structure that makes up the pili. These are similar proteins that we would find in Flagella and Fimbriae for that. The next process we're going to talk about is transformation. So in 1928, researchers demonstrated transformation occurring. So with transformation, we have DNA fragments or DNA plasmids that are in the environment, but they don't have a cell that is containing them, right? So there's no other bacteria involved, but we have a bacteria that's in that environment, whether that is a test tube, media, or like soil or your gut. There's whatever was containing these DNA fragments has died. And so given the conditions, the bacteria will take up that DNA. It can then incorporate that DNA into its genome, into its chromosome. This is not always successful, so sometimes sometimes those DNA fragments just get degraded. Sometimes the plasmid's not even taken up, right? But if the plasmid is taken up, now that bacteria has that plasmid in the genes that are contained within it. You're probably familiar with transformation because you've probably done a transfection lab at some point, either in intro bio class. In genetics, we did a transformation lab also, where you guys had E. coli that had plasmids in them, and you had to do a DNA isolation, and you were specifically isolating plasmids, and then you did restriction digest, and depending upon the fragments you produced, you determined whether you had plasmid 1, 2, 3, or 4. Okay, so again, with transfection, that is a transformation that's being done in a controlled setting in the lab. In the lab, we give bacteria everything they need to grow. We want them to grow. We want them to grow and divide, right? And so there are special types of bacteria that we need to use when we're doing those transfections. So those are known as competent cells, because we know they have the ability to take up, we know from previous experiments, they will actually take up plasmids or DNA fragments in their environment. The other thing, during a transfection, you will probably heat shock your bacteria. So you put them at a high temperature, usually 42 degrees Celsius, like in a water bath, with the DNA, and that just causes the bacteria to be a little more vulnerable. So the membrane integrity is compromised enough that you don't kill the bacteria, but enough that the fragments are more likely, or plasmids are more likely, to be incorporated, taken up by the bacteria. As mentioned in class, when we think about bacteria out in nature, and what we talked about early on in the semester is like what we're doing in the lab is not always what's out in nature, right? So how bacteria grow in nature is going to be different. So many times out in nature, the bacteria are already kind of stressed. They don't have all the requirements they need. It's not the ideal temperature for them, like right on spot. So they are going to be in a more competent state, incompetent in meaning that they're going to want to take up additional factors, additional DNA that might be successful for them to increase their success in survival. Okay. Next, we're going to talk about transduction. So transduction is where we have the transfer of bacterial genes by viruses. So a gene from one bacteria is transferred to another bacteria, but instead of it being direct, or because the bacteria has died and left its plasmid in that environment, it is achieved through viruses, more specifically bacteriophage. So bacteriophage are able to infect bacteria. They will use the bacteria's machinery to make copies of their genome and also express the proteins which they need to make their capsid and other structures that are present in a bacteriophage. When the number of bacteriophage increase, what happens is that bacteria then lyses and releases the bacteriophage to go on to then infect additional bacteria. Okay, so that's a normal bacteriophage life cycle in a nutshell. And so with transduction, when that bacteriophage is infecting the bacteria, part of the bacterial genome ends up getting put into the new bacteriophage that are being produced. I'm just going to go to the next slide. So you can see here how we got the bacteriophage. It has its phage DNA. It then goes and infects the bacteria cell. When the bacteria is now producing the new bacteriophage and assembling it, some of its genome gets incorporated into the genome that's going to be passed on to the next generation of bacteriophage. So when those bacteriophage go and infect the next bacteria, not only is the original bacteriophage DNA there, there's also a portion that is from the original bacteria that was infected. And so it's not always that the bacteria lyses. So at this point, this bacteria would be a recombinant bacteria because it actually has a portion of genome from another bacteria that's been transferred in. So I just left this slide in specialized transduction just so that you are aware there are other types of transduction besides generalized. For our purposes, we'll just focus on generalized transduction. So if you do have any questions about any of the information from this chapter, from this presentation, please let me know. I'm more than happy to answer those. But hopefully this was helpful in understanding some of the mechanisms that lead to genetic variation. Again, please focus on the microbial differences. There'll be questions, some of the background type information, like on mutations. But again, the focus is going to be more so on microbial horizontal gene transfer, which we did that class activity on too. So